I'm Paola Arlotta, and I'm a professor of stem cell and regenerative biology at Harvard University. In this video, I would like to highlight some of the findings from our recent paper entitled DeCoN, Genome-Wide Analysis of In Vivo Transcriptional Dynamics during Pyramidal Neuron Fate Selection in Neocortex, which is a joint effort with John Rind's lab at Harvard University. Now, a distinguishing feature of the mammalian central nervous system and of the neocortex in particular is its high diversity of neuronal subtypes. And decoding the mechanisms that generate and maintain this diversity of cells, has really been complicated by the difficulty of labeling and isolating individual populations of neurons. So here we have developed a high-throughput methodology that combines immunodetection of distinct neuron classes using antibody against nuclear antigen transcription factors with FACS purification and RNA sequencing to define the transcriptional dynamics of 3 neuronal classes which we isolated from early stages of development of the cerebral cortex. We then built an interactive web-based platform which we call DeCoN to enable easy data mining and dissemination to the community. We have applied these methods to the analysis of 3 populations of pyramidal neurons, as you can see here: the callosal projection neurons, which connect the 2 sides of the cortex, the subcerebral projection neurons, which connect the cortex to many different targets below the brain, and the corticothalamic neurons, which connect the cortex to the thalamus. These 3 cell types can be distinguished by their differential expression of a combination of 3 transcription factors: BCL11B, TL4, and SABI2. Now, the schematic here illustrates the steps of the experimental approach that we've taken. First, the cortical tissue was dissociated to a single cell suspension, and the cells were fixed with paraformaldehyde, and at the same time, their membranes were permeabilized by a treatment with saponin. And this was followed by incubation with the selected combinations of antibodies and separation of the 3 neuronal classes using FACS. Then purified neurons were subjected to RNA sequencing to understand gene regulatory events occurring during fate selection. This methodology is scalable, does not require genetic label, and because the neurons are fixed before purification, they withstand a higher pressure condition of purification, which normally preclude the possibility of purifying large number of neurons. And so this method now allows to purify millions of neurons of different classes from the same tissue to allow broad, comprehensive molecular profiling.
I'm Brad Molyneux, a new assistant professor at the University of Pittsburgh. I want to highlight a few of the insights that co-first author Loyal Goff and I made when we analyzed these data. Since we started with purified cells and sequenced the generated libraries deeply to over 100 million reads per replicate, we were able to identify transcripts that are expressed at much lower levels than can be identified with other approaches. Looking at the expression of known genes, we confirmed that the cells of interest were in fact sorted to high levels of purity. With a deep data set and frequent sampling over time, we were able to identify hundreds of coding genes specific to each cell type. And there's increasing evidence that long non-coding RNAs might play critical and varied roles in the specification and function of individual cell types in the brain. But their characterization and functional investigation is really at its infancy, and there's been limited exploration of the cell type specific expression of lincRNAs in vivo. Many are thought to be expressed at very low copy number and are likely missed when sequencing heterogeneous tissue or even microdissected layers. Using our approach of deep sequencing on individual cell types, we identified over 5,000 lincRNAs with specific expression in these enriched populations, over 800 of which exhibited differential expression, often with striking restriction across cell types or during development. As part of this study, we have created an interactive companion website enabling investigators to visualize, query, and manipulate the extensive transcriptomic data, and also can serve as a repository for future transcriptomic and epigenetic data. At both the gene and gene set level, we've provided web-based visualization tools to investigate this data set, including new tools for gene discovery. These data are integrated with existing data from various external sources, including the Allen Brain Atlas in situ hybridization data and processed RNA-Seq reads via tracks in the UCSC genome browser, to facilitate a thorough exploration of the data. The user-friendly interface for analysis at the gene and systems level provides a critical platform for understanding the genome-wide transcriptional dynamics of neuronal differentiation. Looking forward, we anticipate that the approach of purifying neuronal subtypes without genetic labeling will enable more rapid studies in mouse and open up whole new possibilities for transcriptome analysis in the human brain.